Hi everyone, and good afternoon, and good morning to all our colleagues who have joined us today from different parts of the globe. My name is Karishma, and I welcome you all to the second TPS Unconference for this year. Before we begin, I'd like to remind our audience that during the session, all the attendees will be in listen-only mode. If you have any questions for our speakers today, please send it to us via the questions box, and we will address them towards the end of the session. Now, ever since COVID-19 was declared a pandemic, scientists, researchers, and medical experts across the world began their quest to develop the COVID vaccine. The COVID shot is the fastest vaccine to have ever been developed and has come as a ray of hope in our fight against the virus. While there have been multiple theories around the development and the safety of these vaccines, India has seen one of the biggest inoculation drives ever, with more and more people coming forward to get their shot. As per the latest reports from the Indian Health Ministry, vaccinations in the country have topped 100 million, making India the fastest country globally to reach the milestone in just 85 days. With different vaccines being offered by different manufacturers, there's a lot of curiosity and uncertainty about the effectiveness of these vaccines and ambiguity surrounding the age at which they are being administered. We at SNTPS and SN India are very happy to share some light on this topic, resolving our queries, while also busting a few myths about the COVID vaccine at today's Unconference. We have with us today a panel of experts who would be sharing insights on the COVID vaccine scenario in India. Now, on that note, I would like to give a small disclaimer on today's content that will be discussed. It's on your screens at the moment, so I'm going to read it out as well. Information in this panel discussion on the COVID-19 vaccines is medical and health related in nature. The views expressed by the invited experts are their personal opinion based on the latest research, evidence, and advisory issued by the different health ministries and authorities. However, please note that this information should not be interpreted as medical opinion or advice and should not be treated as an alternative to a consultation with a medical professional. For any medical advice related to COVID-19 or the COVID vaccine, please consult your physician. That said, I am now pleased to invite our panelists for today. We have with us Dr. Piyush Jain, who is the Senior Consultant, Pediatrics and Head of Neonatal Units at Hindu Rao Hospital and Associated New Delhi Municipal Corporation, NDMC, Medical College, Delhi. With more than 3 decades of experience as a clinician, Dr. Piyush's areas of interest are vaccinology, infectious diseases, and neonatology, and management of the vaccination against COVID. Along with shouldering teaching responsibilities, he is also the national trainer for the Indian Academy of Pediatrics for Science of Vaccinology. He has been on the editorial board of the flagship journal Indian Pediatrics for 12 years now and is also on the advisory board of several other journals. This journal is co-published with Springer Nature. Dr. Piyush has been involved in Pulse Polio program as well and has also worked as the joint director in Delhi State AIDS Control Society. So welcome, Dr. Piyush. Our next panelist for today is Dr. Dhiren Gupta, who is the Senior Consultant and Co-Director of the Pediatric Critical Care Unit, Pediatric Pulmonology at Sir Gangaram Hospital, New Delhi. Dr. Dhiren has been teaching and training fellows in pediatric critical care and is also the President-elect of Pediatric Critical Care Program at the Indian Academy of Pediatrics. He has received the prestigious Vishishta Chikitsa Ratna Award for his outstanding contribution in the field of pediatric intensive care by the Government of India. He has been the guest executive editor for several journals and has developed a module on conventional chest radiology for practicing pediatricians. He is also involved in the care of COVID patients at Gangaram Hospital and has been steering the vaccine center at this teaching institute. Welcome, Dr. Dhiren. We're very happy to have you with us.
Yeah.
Our panel discussion today will be moderated by our colleague from SN India, Dr. Naren Agarwal, who is the editorial director of Books in Asia and leads a team of editors responsible for developing and delivering the book program in medicine, biomedicine, and life sciences. He has been with Springer Nature Group since 2011 and in the publishing industry for the past 2 decades. A few of his key responsibilities include data-driven evaluation of research output for planning acquisitions, fostering relationships with scientists and research institutes, ensuring compliance and consistency in editorial and production workflows, and resolving ethical issues in scholarly publishing. So without further ado, I'd like to hand over the stage today to Dr. Naren Agarwal to begin today's discussion with our panelists. Thank you, and I will see you at the question and answer round.
Thank you, Karishma. And one more time, I would like to extend a very warm welcome to our invited experts, our speakers. And before I go ahead with the question, I would like to uh, share one important thing here with you that many of you might be asking, that COVID vaccine is for adults. Then why both the experts invited here today are pediatricians? And it's an obvious question. The reason is this, that pediatricians across the world are the real vaccine experts. They are the people who vaccinate all the children on this planet. They have the widest experience of dealing with vaccines, dealing with a widest possible variety of vaccines. They are the ones who have dealt with all the side effects, and they are the experts who work with the policymakers and societies to determine that what should be the dosage schedule of a vaccine. So, keeping that in mind, we decided to invite the pediatricians who should be able to share with us their opinion and views and expertise around certain questions that are all important to us. So, the structure of this discussion today here is that we will be circulating the questions alternatively, and first speaker or first guest would be invited to reply to the question, and any additional information will be extended by the invite second guest. And then we will put the next question to the second guest. This is how we are going to circulate the question. These questions have been discussed and curated beforehand. After we have covered a certain number of questions, we would make the house open and we'll start inviting questions from the audience here. So Dr. Piyush, Dr. Dhiren, welcome once more. And I would like to begin with a general question that tell us something about the variety of vaccines that are currently available around the world. We keep hearing a lot of things about kill, live, genetically engineered. We wouldn't really want to go into very technical details here, but still we would like to have some idea that what do we have here in India as compared to what is being offered to people outside India. Question to you, Dr. Piyush.
Thank you, Naren, and a good afternoon to all the audience also. Excuse me. As we are talking about the COVID vaccine, I'd like to start with COVID vaccine only and then come out to other vaccines if the time permits. The COVID vaccine, as any other vaccine, would give immunity against the coronavirus and the COVID disease, COVID infection itself. And there are about 9 to 10 types of technology platforms on which the COVID vaccine is available around the world. And most of these platforms you would have heard and would have read in the papers are built towards the use of the spike protein antigen, which is present of the COVID vaccine or the COVID virus itself. I'll start with the basic platforms or the technology which we have. The first is the RNA vaccine. You would have heard something about the mRNA vaccine. These are the Pfizer vaccine or the Moderna vaccine are these type of vaccines. What happens is that mRNA is a kind of a code which any virus or a bacteria makes, and this when enters any human cell or tissue, then specific protein is formed, and this protein acts as an antigen, and against this the antibody would be produced. So this is how the mRNA vaccine works. The mRNA of the virus, of the coronavirus, is introduced in the human body. The mRNA enters the human cell, and the proteins would be formed, specifically the spike proteins will be formed in the human being, and this will act as an antigen. Also are the adenovirus vector vaccines. These are the some things which you have heard of, which are the common ones are the Oxford extragenesia vaccine, the Covishield vaccine which we are talking about, even the Russian Sputnik V vaccine or the Chinese Covidia vaccine. They're all type of the adenovirus vector vaccines. The new virus vaccine which is coming up is Johnson & Johnson. This is also an adenovirus vector vaccine. What happens is that although coronavirus is a virus, but if it is introduced into the human body, it will cause the disease. So what is being done is that a part of the coronavirus DNA is taken, and this DNA is introduced into some adenovirus, and that adenovirus is introduced in the human body. What happens is the adenovirus start making certain proteins from the DNA of the coronavirus, which is incorporated in that adenovirus. These proteins will again act as an antigen, and antibodies will be produced in the human body. And this is how the adenovirus vector vaccine works. Also, some kind of inactivated vaccines are also available. Inactivated means that coronavirus is taken, it is cultured or prepared in the lab, and then it is killed with the use of substances and chemicals like formaldehyde. So once that virus is killed, it is then introduced again in the human body as a vaccine. What will happen is it will not replicate. This killed virus will not cause the COVID disease, but will form but the antigens present on the surface of the virus will act as antigen, and again antibodies will be produced, and this will act as a vaccine. Also, there are certain other type of vaccines like subunit vaccine. In a subunit vaccine, only a part of the virus is taken, and this is introduced in the human body. Something like a Epivac corona vaccine or even a Novavax vaccine is a type of subunit vaccine. These are not yet available in India right now, but these vaccines are also a type of vaccines which is used in many other viral diseases also. I told you there are about 9 to 10 platforms. These are the 3 or the 4 major platforms on which we are working right now for coronavirus vaccines. Other platforms are something like a virus-like particles. There can be a DNA, say, plasmid vaccine. This is just to complete a list. Instead of adenovirus, you can use other virus as a host or a vector. That can be a lentivirus vector or a conjugate vaccines, which are some other vaccines can also be used. For example, a meningococcal conjugate vaccines. This is just to complete the list. Right now, we don't have a conjugate vaccine for coronavirus, but they are being produced and probably might be available in a couple of years. The last vaccine which is available for coronavirus and is still being studied is a vesicular stomatitis virus vaccine. Again, this might take years to come up. These are the vaccines which are available around the world for coronavirus. But the most important thing for us is what are the vaccines which are available to us in India, and what more vaccines can we look forward to in the coming months. I'll just take a couple of minutes and enumerate them. The first is the Covishield vaccine, which is made by the Serum Institute in Pune. The second one is the Covaxin vaccine, which is made by the Bharat Biotech Hyderabad. You know everything about it, and we'll discuss it in the coming hours about these vaccines because these are right now available in India and available to all. The next one is the Sputnik V vaccine, which is being made by Dr. Reddy's lab. And actually, the government of India gave emergency use authorization for it to be used in India only a couple of days ago. So it is likely to be available in a couple of months for us Indians also. Also, the Johnson & Johnson vaccine is being made in collaboration with Biological E. This is a company and probably would be available to us by the end of August. Also, the Novavax vaccine is made in collaboration with Serum India. Another couple of vaccines which would be available probably by August or September are the Zydus Cadila vaccine, and also you might be interested in knowing that an intranasal vaccine is being developed again by a biotech, but this might be available by next year or so.
All right, thank you, Piyush. Dr. Dhey, any quick comments?
Yeah,
Very brief
I
comments.
think, I think it's, if we just try to simplify, Dr. Piyush has wonderfully explained. It's a very comprehensive answer. Remember, we want an antibody against the active component of virus. Fine. The active component of virus is a part of S1 protein. S1 is a spike. So that part is RBD. Your this RBD, that is receptor binding domain, and if you further go into, there is a RB moiety, that is a sub part of that domain that actually binds with ACE2 protein of our body. So if any vaccine, we are able to produce antibodies against that subpart, which is actually responsible for binding to ACE2 protein, we call it effective vaccine. Why so many platforms? The problem is that configuration of S1 protein changes. Fine, when it try to bind. So we want before it changes and leads to binding with the with the human cells. Just before that, it exposes its antigen. Just before that. Fine. So it is something like you must have seen the tortoise. It's in the shell. So when you touch it, is inside the cell. Then suddenly, gradually, it takes out its tentacles. So same way, this protein S1 takes out its tentacles. That is a brief time. At this brief time, we have to you can say freeze that moment when it exposes its RBDs, and that frozen movement has to be introduced in the body, human body, to produce antibodies against it. So that is a problem, to stabilize this frozen movement and to make it enter in the human body. So that is the biggest challenge. And another problem is that the mutation, which we are going to talk subsequently. So in nutshell, we want a antigen, a complex antigen frozen at a moment when it was exposed, a weak moment of, you can say, that S1, and we want that to be enter in human body safely and doesn't interfere with normal human functions, and that we call a vaccine.
Okay, great. Thank you so much, Dr. Dhiren. Can we have the next question, please, on our screen? This is like an extension of the first question, Dr. Dhiren. We hear that maybe some vaccines are kind of a gene therapy. Maybe
Got the
the
point.
mRNA vaccine.
Act,
What are
Actually,
they?
this is a misnomer that your mRNA vaccine enters the nucleus. No. So I got a human, I'm a human, I got cell. Okay. In cell, there is a nucleus, there is a DNA. Okay. Now this DNA and we have got mRNA. The DNA convert into mRNA and makes the proteins. Fine. So we are bypassing that cycle. We are just introducing the mRNA, which is surrounded by some lipid membrane. That's why you need lot of minus temperature of -70 to stabilize the things, like in Moderna and Pfizer. So that is placed in the cytoplasm. That has nothing to do with human therapy. So it's a misnomer using the word gene therapy. So it has nothing to do with modification of my gene. So my progeny is not going to change as per the virus mRNA. Fine.
All right. That's very reassuring that it's not a gene therapy. Thank you so much. Can we have the next question, please? Dr. Dhiren, the
Yeah.
next natural question, the obvious question would be, how safe are these vaccines? And if they are,
Uh,
then we keep hearing about several countries pausing the usage. Recently, we heard about USA pausing the use of J&J vaccine, which is a single-dose vaccine. And we are hearing about
yeah.
reports here and there about blood clotting. And all of these side effects are around blood clotting only. So what is happening here?
Yeah.
Can we have a quick answer from you on this?
Yeah. Naren, what is happening? The policies has been politicized. Same way, vaccinology, lot of politics has entered. So that is, and premature release of the data from the competitive sites has actually been confusing on the websites and the social medias and all that. So one thing is very clear. We subject our kids for the vaccination. Influenza vaccine can cause Guillain-Barré syndrome. So many vaccines have got so many side effects. Still, we subject because we are subjecting somebody else to for the vaccination. Now, for the first time in my lifetime, the actual adults has been subjected. So we are too cautious. Means now we are thinking too much. Okay, what are the major side effects? What are the minor side effects? And as compared to pediatric, we are more reactogenic. Pediatric age group is less reactogenic. Second,
Okay.
we have to weigh the balance between the benefit and the drawback. If you have seen any COVID patient and actually you land up in pneumonia, and means for months together, they land up in major crises. Not only heart, not only lung, also brain and intestine, and means their life is spoiled, most of the patient. So, so we have to weigh that nothing comes free in the nature. We are interfering with the nature. So nature is going to produce some side effect. When we are introducing S1 protein in the human body, so that is not going to going to come inside without any pay. We have to pay the price. Fine. What is that price? If the price is outweighing the benefit of the vaccine, okay, we won't purchase it. But when we find that, okay, the benefit is outweighing the price we are going to pay in form of side effect, okay, go ahead. Now, what is that price? That price is in form of local side effect. That price is in form of fever for 2 days, 3 days, headache for 2 days, 3 days. I think that all is fine. Now, the problem is happening is that few deaths have happened, and that is implicated to the clots. Of course, it is now means it is in NMJ, means NJ, New England Journal. Now it is published that okay, these few patients were predisposed to the clot formation, and it led to the brain clot formation and led to lot of complication. Now, if you remember what is happening, FDA trying to find too much fault in AstraZeneca trying to find in the Covaxin, and Covaxin trying to find fault between, these things are happening, and this has made the confuse. So if we talk about science, presently, I'm talking about present situation, the number of acute thrombotic event is actually very less. See, we have given this injection in crores. Till now, we have given injection in 9 crore. Suppose 180 of the patients actually land up in some complication which was derived, which was initiated by the subunit vaccine or any vaccine. So 180 out of 8 crore, you don't know whether actually this virus led to this problem, or even if we assume that this virus was the initiator of prothrombotic state, still it outweighs the benefit of the vaccine as compared to the side effect. So even if we assume that everything is true, we find a 180 patient died within 72 hours in India. Out of 8 crore, 180, I feel that these patients must be predisposed for prothrombotic events. There must be some other factors which led to death. Probably we need more monitoring. We cannot accept 180 deaths also. That may be the wrong message, but we want to be more precautious to have more precautions in the future. But this vaccine program should continue. There should not be any confusion among those people who are going to get this vaccine regarding the benefit of the vaccine at present date.
All right.
So let me, I'd just like to intervene and just tell you a couple of more things. What is happening here is what we talk about is called as a VIPIT, vaccine-induced prothrombotic immune thrombocytopenia. Actually, these are the type of clots which happen with many other conditions also. The figures right now are that if you give about 1 to 2 million vaccines, and these are happening only with the adenovirus vector vaccines, like, for example, Johnson & Johnson, or with AstraZeneca and the Covishield vaccine, and probably with the Sputnik one also, which we don't know about. Then the incidence is only about 1 in 1 million or so. Whereas the incidence of the same type of a clotting, which is seen in females who are taking contraceptive pills, is more than 1,000 per million doses of contraception. So you can see that 1 in 1 million due to vaccine is just nothing. So we should not even think about the benefit outweighs any of such side effects. So we should, I think, just forget.
So the message would seem to be that by and large these vaccines are safe, and a kind of vigilance or regular continuous monitoring is required to really assess the incidence and reasons for the side effects of these vaccines, and that is a job for the regulatory authorities and scientists that they are looking into. And by and large, these vaccines are safe to take, and the benefits really outweigh the risks associated. Thank you. Can you have the next question, please? This is an important question, and we keep hearing about the efficacy, and it has come to this point that we know more about the efficacy percentage of a vaccine more than we know about the mileage of a car that is available in the markets. So what is really happening here? 60 to 95%, and naturally all of us would like to go for the best possible vaccine, the one with the highest efficacy. What does it really mean? Is it that 30% people will not get the protection, or 50% will not, or 5% will not? Quickly, for you, Dr. Piyush.
Actually, what we are talking about efficacy means that it means the reduction of the disease in those who are vaccinated compared to those who are unvaccinated. So
Okay.
what we have to look at is not only the effectiveness of the vaccine, but also the conditions in which the vaccines and the unvaccinated people live, or for example, social distancing, or whether they follow the COVID appropriate behavior or not. So basically, this is kind of a statistics which have various connotations in various areas. So also, what is important for us is, as you just told us, the vaccine efficacy varies between 60 to 80% between various vaccines. But also, what is important for us is what is this efficacy is for. This efficacy, I like to tell you, is not for infection. This is for disease. Means that suppose we say a vaccine has an efficacy of 70% towards COVID, it means that it would decrease the serious COVID disease in a patient who have taken vaccine by the tune of 70%. It does not mean it will stop the infection from being occurring in that patient. So
Okay.
right now, whatever figures we have is the infection is reduced. No figures are available as a just a ballpark figure. Infection is being reduced only about 20 to 30%. For example, if I am vaccinated, if I have taken if I have taken 2 doses of a vaccine, that is a complete immunization, and then also I have had those doses 2 to 3 weeks ago, that means I should have developed antibodies by now. Even then, I have a chance of getting infected. Getting infected means the virus would have entered my throat and I would be able to spread it to various persons. I might not be getting the serious disease. I might not go getting the serious disease, and that I might not be hospitalized for COVID, but I'll still have the infection in myself. So the vaccine is not 100%. It does not prevent that 100% disease. It prevents, it does not prevent the infection. It only prevents the disease and the serious disease from occurring. And also, these efficacy figures for various vaccines varies from place to place and from in various studies from 60% to 80%. Why this so is also because the various vaccine manufacturers and various studies take the definition of disease to be different. Infection is the presence of a virus in the throat, which is taken care of, which is identified by the use of RT-PCR, where the disease is identified by the presence of infection and the presence of certain symptoms. One vaccine has taken the presence of one symptom as a disease, another study might have taken as 2 or 3 symptoms as a disease. So actually these efficacy studies are somehow not comparable to each other. So again, the jury is out. We would not be able to comment on this right now. Also, what is important is whatever vaccines is available right now, we have to take them.
All right. Okay. Dr. Dhiren, would you like to weigh in with any quick comments?
Yes. Now, what is happening? As I told you, there's a competition among vaccine makers and the countries now. Fine. So if we talk about science, all these efficacy studies, whether it was endpoint of the disease, whether it was an endpoint of nasopharyngeal swab being positive, these were done few months back when the virus didn't mutated. Fine. So if we talk about present, what Dr. Piyush said is correct. Presently, in present scenario, what type of viruses we have in our community, we should know. And secondly, we should look into the present context. Like for an example, if you find Moderna vaccine, so there are two components of infection. One is the disease. Disease has got two parts, mild to moderate disease and serious infection that requires ICU care. Second part of the disease is nasopharyngeal carriage. Like even I'm vaccinated, I can still carry my virus in the nose and can become spreader. Even if the virus is not able to penetrate my body, even if it is able to penetrate the body, but not able to induce serious disease. So there is a spectrum of the disease also and infection. Now, if we talk about Indian vaccine, our vaccine is not able to prevent the carrier state and low grade asymptomatic infection as compared to what they have proved in Moderna. It was found to be 61% reduction in the carriage state. Because when they studied the Moderna and the Pfizer couldn't study that, but Moderna studied. They took the sample before the next dose of the vaccine, second dose of the vaccine, and they found it was reduced. Nasopharyngeal carrier stage was reduced. So one is the carrier, or you can say asymptomatic carrier. Now we come to the disease part. Disease is two types: mild to moderate and severe disease. Now, when we talk about mild to moderate severe disease, our vaccine and their vaccine means Pfizer and Moderna has got efficacy of 95%, and Johnson and Johnson has got efficacy of say 60 to 70%. But those studies were different in different eras. When the Moderna and Pfizer studies were done, then mutation was not there. When the Johnson and Johnson was done, the mutation happened in like South African strain. So that's why efficacy is different. Now, if we talk about our vaccine, you must have heard stories that even in our hospital, in many hospitals nowadays, there is no hospital where there is not an outbreak of infection in doctors. Okay, why it is happening? There are two things. One is that vaccine gave us some confidence. Okay, we can be little bit relaxed about the norms of because everybody was tired, especially when they were eating together. They were at home. They were not as following the norms as it used to be last year. Second, probably our vaccine. Already our vaccine, the Covishield and Covaxin, they said that our efficacy is this much. So it means that already efficacy is less by 30%. So we are only able to prevent 60 to 70% of infection. Which type of infection we are going to prevent? That again depends on what type of mutation we have. As per my knowledge, 100% of the viruses in Delhi has been mutated presently. Fine. The original strain is no more there. So there are two types of mutation. One is your UK strain, second is your South African strain. Suppose we are 100 people listening to this webinar, and 100 of all get infection in due course. Unfortunate, I'm not saying, I'm just implying. Say they get infection in next 15 days. Fine. 70% may get an UK strain and 30% may get South African strain, and all were vaccinated by Covishield. So you may find that those people who got vaccinated by or who got infection by your the London strain, this vaccine was able to save that serious disease. But regarding South African strain, probably our vaccine won't be able to prevent the serious one also. Because yesterday there was a study published, they said that E454K, that is a part of double mutation, that is a predominant mutation in South African strain. Even Moderna and Pfizer sera, which they collected, was unable to mount antibody response for virus replication. And forget about Covishield, which is much inferior, I can say, than those vaccines in mounting the antibody response. So that's why only one thing I will say with certainty, that is the mask. Don't rely on any vaccine. We have missed the bus. We are pretty late in vaccinating the people. Get yourself vaccinated. Get yourself whatever is available. Dr. Piyush has rightly emphasized. Get yourself vaccinated. But don't rely on the vaccine. That is very, very clear. And I'm doing counseling of adult, and almost means I am not getting time for washrooms also. To get into washroom and see, it's so difficult. It's so rampant. People who got infection in November are getting reinfected. People who got vaccinated are again getting infected, and many of the patients are landing in severe pneumonia also, those who are vaccinated. Every week, my version is changing and the type of patients we are getting.
Okay, so the message is clear. Get vaccinated, but keep wearing your mask and stay at a safe distance, right?
Yes.
Precautions are still more important than putting our faith into vaccine till we have more data and till we know more about how these vaccines are going to help us. At the same time, it seems that these vaccines certainly prevent a serious disease in people who are taking these vaccines. So can we get on to the next question? At the same time, I would like to say here that we are left with 20 more minutes. So we will be taking two more questions, and maybe we can, maybe it would be best if we don't have a lot of technical details coming into the replies. That would be very helpful, Dr. Piyush, Dr. Dhiren. So this question goes for Dr. Dhiren. I think partly you have covered it, that people are getting infected even after getting 1 or 2 doses, and you have explained the
I think
reason for
I have answered this.
yeah. So it is not a vaccine failure. Can we say that it's not a vaccine failure, but it is just the nature of the virus, that despite having a vaccine, maybe you are getting protected from a serious disease, but it is not going to prevent infection. Correct?
Yeah, virus is becoming smarter than the vaccine, so we need to be smarter, have an extra coat of precaution, that is mask.
Okay, great.
Also, it takes the vaccine to act. Antibodies will be formed at least 2 to 3 weeks after the second dose. The patient,
Sure.
if he gets infection before that time, would always the vaccine. We would not say the vaccine is not working. We haven't given it the vaccine
Right.
to be a specific time or enough time for it to work.
Absolutely.
That's
Absolutely.
what it is
So,
nowadays.
can we see the next question on our screens? Thank you. So this is a little tricky question. I read somewhere that during the vaccination phase, immunity in general is down. Your body is focusing on dealing with the antigens and trying to produce antibodies, and actually your immunity is going down. Is there any such link that when you get vaccinated for a certain period of time, your guards are lower and your immunity is really taken a hit and you can fall ill during that phase?
Absolutely not.
Absolutely no.
Absolutely no. Okay, great. That's a big relief. And can we have the next question, please? Important question. Does alcohol affect the efficacy of vaccine?
Absolutely no. It
No.
doesn't affect at all.
Are
Not
you
with,
saying that there is
not
a
with the
safe limit? Are you saying that you can have it? What is it like?
you can continue to have the same amount. The only thing is the alcohol, if taken in the larger amount, will decrease the immunity as such. So it's not a good appropriate COVID behavior to take a lot of alcohol, but it would not have effect on the vaccine.
Okay, so in a moderate amount, no effect on the efficacy or immunogenicity of the vaccine. That's very reassuring. Thank you, Piyush. Next question will be for Dr. Dhiren. Can we have the next question, please? If one gets infected after a few days of getting the first dose of COVID-19 vaccine, should we delay the second dose? Do we really need the second dose? Because if the infection has happened, body is still going to make antibodies. So is there still any relevance for
Yes,
going to the next?
science, yes, Naren, science is evolving. So for present sake, if you get infection, wait for another 4 to 6 weeks, then get a second jab. So that is the present, you can say, recommendation. But of course, every week the things are changing. So second, where the infection which you got probably may act as a booster, especially if it was a symptomatic infection. So I think presently people are getting infection. Another 4 or 5 weeks you may find there may be change in the recommendation, and but again, as I told you, means vaccines are just one protective measure, but you need to continue to wear mask, even if you are got infection with some of the bug, because there are many viruses which are moving around. If you get one bug, you can definitely get another bug another month. So no protection is guaranteed, whether it's a natural infection by one mutated strain or by vaccine.
All right. Great. Thank you. Very helpful to know this, Dr. Dhiren. My request to all the audience is that now they can start sending in their questions to Karishma in the chat box. Yes, and in the meanwhile, while she is about to receive questions from you, we can have a go at another question. Can we have the next question, please? We have come across this association between C-reactive protein and getting side effects, serious side effects on the vaccine. And I have had a lot of friends call me up that should we get the CRP done before we go for vaccine for our parents or ourselves? So is there any correlation, is there any causal linkage between a level of CRP and side effects, potential side effects of a vaccine?
Absolutely not.
Question to you.
No need to get any investigation before a vaccine. Vaccine has to be taken. That's the issue ends here. Has to be taken, and appropriate behavior has to be followed, including masking, as Dr. Dhiren said. That's all.
So there is no recommendation by any scientific body or regulatory authority or the vaccine manufacturers that you first get your CRP done before you should go for the vaccine.
No, one point I want to make. Yeah, one
Yes,
point
Dr.
I want to make, that when you're preparing yourself to get a jab, just see to it that nobody should be suffering from infection in your society, in your house, because it may be possible that you are having this infection during incubation period. You get a jab, another 2 days, you actually get a symptom of the active infection, not the jab related, and you start feeling okay, the rise of CRP, and then you start spreading the rumor that look, I got vaccine, and following vaccine, my test came positive, so it may be that test is related with the vaccine and side effects are related with the vaccine. No, please, it is no relation with the CRP, but of course, it may be the active infection during pandemic. There's a massive spread presently. So likely you were carrying the infection, you were in incubation period, you got a jab, and the symptoms mix up post-vaccine.
Very helpful advice. And again, to all the attendees here at this event, that for any questions related to vaccine towards taking a decision in for your own health, please consult your own physician. Please do consult your physician for these answers. Karishma, do we have some questions from the audience, or do we have time to take the next question as well? It's already
I think we should move to audience Q&A, because we've got some really interesting questions which have come in.
Great. So all right. So Piyush, Dr. Dhiren, now you can expect some questions directly from the attendees, and Karishma would be helping reading out those questions to you. And Karishma, you can follow the same rule for putting these questions alternatively.
Absolutely. So the first question is, how is a dead virus used in the vaccines, since the virus is not live until it enters the host? So how does that work?
Actually, what happens? Happens? Any virus, any antigen, which I told you that is a S spike, if it has to enter in the body, we need one more thing. That is a stimulant. We call it alum. Okay, like a adjuvant, and that adjuvant stimulates the immune system to produce antibodies against that, because that is a dead one. Okay, one is that it's a good number, it's a huge number, huge amount that is going to enter the body, and we also have got some adjuvant along with that. That adjuvant makes the immune system little bit hyperactive to act against that protein which you have entered artificially. So is something like we are stimulating the people. We are not just passing the knowledge like in 100 of the group. We are not only passing the passive knowledge, but we are actively stimulating. So it's a oldest technique, that is the dead virus, and mixing it with the adjoint and then making it active when it enters the body.
Okay, so that's the
What has happened is that
technology of science.
you would have heard of the injectable polio virus, the polio vaccine. The injectable polio vaccine is also built on the same principle. It's a dead polio virus. Polio virus is killed. It's called as inactivated and then injected in the human body. And we have been using this injectable polio virus, the polio vaccine, for years together. In fact, this is one of the best type of vaccines which are available because the side effects are very less in the sense that there are no chances once the virus is killed. There are little chances of it being activated once in the body. So it has lesser number of side effects and is proven to be very effective. So the same thing is likely to happen with the coronavirus inactivated vaccines also, because this is a tried and tested technique for years together.
And especially for immunocompromised, those patients who are on immunocompromised, this is a time and tested method.
Thank you. The next question is, are the sample sizes of the efficacy studies similar? And can we compare the efficacy of one drug to another, or rather one vaccine to another?
I think Piyush sir has answered, very well answered. Sir, repeat kar do. You can repeat.
What I told you, because the sample sizes of the efficacy studies can be compared only if the endpoints are similar. What happens in various studies, because there are different manufacturers, the studies have a different parameter, the methods are different, and also the endpoint which they are studying are also different. So it's very difficult to compare these studies at the present. So we can say
one point I want to make. What happened? For an example, when Covaxin vaccine had phase 3 trial, there was a lot of hue and cry. Why not they are not showing their data? Why not they are not showing their data? And we have got approval. What happened? They had around 25,000 people who got the jab. But at that time, your pandemic was weaning off. So they were not getting that amount, that number of people, that number of events in those people. Fine. So probably that's why it took time. So not only the number of candidates matters, but when it was done, at what time it was done, and at that time what type of virus was in circulation. So there are many factors. So that's why it is not just the numbers. It is the other factors which affects the efficacy that we need to understand.
Great.
Okay.
Thank you. Karishma, please go ahead.
Sure. The next question is, regulatory bodies have now allowed other vaccines for emergency use in India. So don't these vaccines need any clinical trials before they are put to use to the Indian population?
Yes, they would be needing.
Sir, I'll answer then. Dr. Piyush is expert of this. I will try and he will add on. See what happens. Suppose like we talk about Pfizer. And if you talk to your friends who are Indian actually in America, and they must have crossed 1 lakh, the number must have crossed 1 lakh. Fine. So previously we used to say, okay, in our population with our genes, was it tried? No. In this one, in the present state, we gave authority to lot of like Covishield, Covaxin, and usually it used to take 5 to 10 years, and we have finished up that process within 5 to 6 months. So we have compromised on that. Fine. And now it is a modified compromise. Like a Pfizer or Moderna, these vaccines has been well tried. Janssen has been given to 8 million people in US. And there must be more than 50,000 Indians who must have received. So what we can say, they were safe. If company produces that data, okay, these were safe. The safety data it produces. Then probably what Indian government has done, Dr. Piyush is better in this field. that they have asked, okay, inject in around 100 people over 7 days. Look at any complication. If no complication, then produce your data and you are free to inject. So I think there is nothing wrong in that. Already we have missed the bus. Very likely all these vaccinations are going to be futile. The way the things are changing quickly, it should have been done 2 months, 2.5 months back. But there were limitations from the government side, from the vaccine manufacturer side, and mindset of not only general people, also of healthcare workers. They didn't came forward to get a jab. Lot of rumors in the social media that has actually led to harm, and probably we could have saved this third wave. And but what is destiny? I think it's very difficult. Virus is much more smarter than us. Sir Piyush, sir.
Dr. Dhiren has explained very beautifully, nothing more to add. Only thing is, as he has said, if the vaccine is good enough for the half of the world population, it is good enough for us. And that's the same thing has happened with other vaccines also. All those vaccines we took about 8 to 10 years, even more, just to testing and testing. And finally, they were all good enough for Indians. Right now, we don't have time. We are short of vaccines effectively. We need more vaccines. So if they are available in any place of the world, and so India government has done the right thing in giving them whatever emergency or whatever permissions, whatever they can, they've started us, and the vaccines will be coming to India within a couple of months. So this is the way forward, and we have to arrange for these vaccines and make them available for Indians. Nothing is wrong.
So no, yeah, Dr. Piyush, one thing I always say that if you go to a restaurant, if you find the owner of the restaurant is serving food to his kid in that given restaurant, it means you
Obviously.
are pretty safe. So if it has been given to million population in US, and that they take care of their population much more than what we do, that is very truth. And I think we are pretty safe. That thing one should remember.
Absolutely.
Okay,
Well said.
great. Karishma, do you have time for one quick question? We can't
Yes,
stretch
we do.
beyond, it's already 3:56, so maybe 2 more minutes and then we can conclude this session. Yeah, please go
So
ahead.
the next question is, how long will these antibodies last in our body or protect us from getting the disease once we're vaccinated?
No
Piyush.
one knows. No one knows. Absolutely, right now, the vaccine program was started only about 5 to 6 months ago. So we have the data for all those only 6 months. But looking at the old other vaccines which are prevalent in the world, the virus vaccines, it is likely that these vaccines antibodies are, if you follow this is a virus, and we have vaccines against other viruses also. So all those once vaccinated, the antibodies usually last for years together. And also the type of antibodies which we are looking at is not only the ones against which we can do a testing. There are also the T cell immunity which is produced from these vaccines, which are not measurable through any test right now. So what we should look at is the protection against the coronavirus rather than the presence of antibodies. And regarding whatever data we have available is that second, the repeat of any of this vaccination is right now not required for any of these vaccines. But also, I like to put a rider that they are people are and the experts are now thinking of having a booster of Covaxin or some other vaccine after 6 to 8 months. So actually, the studies are going on, and we'll be able to tell you probably as the results come on.
Thank you.
Okay,
Yeah.
Dhiren, would you like to add?
I think the flip side is that even if you have got antibodies, you can have infection. Second, is a T cell immunity that also plays a major role. But regarding Covishield, it has been said that the maximum T cell response the people get within 4 weeks. If you give another jab, that is a booster, doesn't add on to the T cell. So what we are viewing that despite of getting Covishield, people are getting infection. So what is happening? Probably for that virus strain, you have got good immunity, but some other virus strain is producing the infection. So very future, in near future, like in influenza, you will have vaccine which will have combination of many mutated strain. Like in influenza, we get 4 mutated strain in one go. You inject it, and and the latest one will be introduced, and the oldest one will be deleted. So this way they have proceeded. Every year, 2 types of vaccine we get. Same thing is going to happen in the this vaccine, coronavirus vaccines. So what we'll have? We'll have Brazilian strain, we'll have UK strain, we'll have South African strain, we'll have Indian double mutant strain in one go, and that will give us some protection. So presently, nothing is sure that the vaccine you are going to get will save you. So probably vaccine you should definitely get whatever the best vaccine available to you. But along with that, don't leave the best vaccine, that is your mask.
All right. Great. So as we are nearing the end of our session, I would like to just quickly summarize that irrespective of the vaccine that you are getting, get yourself vaccinated when your turn comes. By and large, they are effective and safe within the explanation provided by Dr. Piyush and Dhiren, that what efficacy means and what kind of protection do these vaccines actually provide. The virus is evolving, and so are the vaccines. They are also going to change in future, and we are going to have a lot more data available to us, so that we will have more clear answers to several of the questions that were raised here today. So with this, we come to the end with the one clear message: there is no replacement yet to masking and social distancing. That is extremely important. And with this, I would like to thank our guest, Dr. Piyush. Dr. Dhiren, thank you so much for joining us. And to all the attendees who have stayed with us through this event, and they asked some really smart questions, which I hope were useful. And I hope that by and large, the answers that you got from the experts here were of some benefit to you. And of course, if you have to further seek any answers, please get in touch with your physicians. Thank you once again, and over to you, Karishma, for final thank you note.
Thank you.
Thank you,
I'll take
Narendra.
a quick minute to wrap up. Yeah. So I'd like to thank Dr. Piyush and Dr. Dhiren for joining us today and sharing their insights on the COVID-19 vaccine, as well as Dr. Naren for wonderfully moderating the panel discussion. We still have quite a few very interesting questions which have come, which we will share with you. And we request you to share your thoughts on these, and then we can send it across to our colleagues who've attended today. A big thank you to our entire audience as well for joining in. We look forward to seeing you at the next Unconference, where we'll pick up yet another interesting topic for discussion. So keep an eye out for the announcement on Hive. And till then, thank you so much and have a great day ahead. Thank you.
Thank you, everyone.
Good evening. Bye.
Thank
Take
you,
care.
everyone. Thank you, Dr.